Welcome to today's lecture on molecular interaction networks. Please don't distribute. So in today's lecture, you want to make sure that you understand what the different types of networks people typically assay in molecular biology are, as well as their pitfalls behind or limitations behind how they're constructed. You want to be able to make sure for genetic interactions specifically that you understand how people generally infer information about gene essentiality or pathway essentiality based on single and double mutant experiments. In terms of general network structures, later on in the lecture, we'll talk about how to infer measures of gene importance based on just network structure and connectivity alone. So you want to make sure you understand how to measure those different types of importances. You want to make sure that you understand what different types of structures in the network represent. So for example, we'll talk a little bit about how to identify communities of nodes in graphs. So you want to make sure that you understand what a community represents and how do you find them and things like that. We'll talk a little bit about, we'll talk very briefly about problems related to systems biology, which is a field basically concerned about, it's concerned with predicting the behavior, the sort of very precise behavior of transcription of different target genes based on assumed regulatory logic. So basically it's concerned about what the effect of having multiple repressors or activators acting on a single gene's expression level is if they're all kind of working at the same time, and how do logic gates affect or teach us about how gene expression can be controlled by multiple regulators. And finally, you want to make sure that you understand what it means for a network to be scale-free or modular. And so in today's lecture, we'll mainly be covering regulatory protein-protein interaction, co-expression, and genetic interaction networks, because those are quite frequently assayed using sequencing-based technologies. We won't really talk about metabolic or signaling networks, although you should be aware that they do exist. But basically, at a super high level, regulatory interaction networks are networks where nodes represent either, for example, transcription factors or some kind of transcriptional regulator, as well as the target genes that are being regulated by those regulators. And so those graphs are networks. So in this lecture, I use the term graph or network almost interchangeably. For the purposes of this class, they mean the same thing. But these graphs are what are called directed graphs. And so what that means is that the edges in these graphs they have direction to them. And so generally speaking, in a regulatory network, edges start from a regulatory node like a transcription factor, and they point towards a node that is being regulated by that regulator. And so that would include basically the target gene. In a protein-protein interaction network, those networks represent actual physical interactions between protein domains or protein complexes even. And so individual nodes typically represent individual protein domains or whole proteins, and an edge between a pair of nodes represent some kind of physical contact. So typically there's like some complementary protein interaction interface on both of those domains, and that allows them to basically physically interact and bind to each other. In a genetic interaction network, those networks are a little bit more interesting in the sense that nodes in those networks represent genes, and edges between the genes, edges between a pair of genes indicates that there's some kind of non-additive interaction between them. And so I'll explain what that means in a few more slides. But basically what that means, for example, is that if you were to knock out each gene individually and assess some kind of phenotype, and then do a double knockout, for example, the phenotype of the double knockout is significantly different than what you would expect based on the phenotype of the individual single mutants. Finally, we'll talk about co-expression networks in which a again, the nodes, each node represents a single gene in the genome, and a pair of nodes is connected if that pair of genes tends to be co-expressed across a number of conditions or samples. And so what that means to be co-expressed is that if one gene is relatively highly expressed in a sample, then the other gene is also highly expressed. Or if one gene is poorly expressed in one sample, then the other gene also tends to be poorly expressed. And so edges in a co-expression network kind of track which genes tend to be turned on or off at the same time.